Today I'm going to discuss about the duck plague virus. This virus is also known as duck virus, enteritis virus, and this is also known as enacted herpes virus 1. And this virus belongs to the genus Mardivirus of the subfamily Alpha herpes virini in the family Herpes viridae. And this virus causes one of the important disease of the duck known as duck virus enteritis, which is a major contagious and fatal disease of duck, geese, and swan. And this is having a worldwide distribution, whereas the migratory waterfall plays a crucial role in the transmission, and also horizontal and vertical transmission plays a significant role in the disease transmission. And this virus is having different field strains, and which differ in virulence. And like other herpes viruses, this virus also undergoes latency, and the trigeminal ganglion seems to be the latency site for this virus. And when this virus get reactivated, they can cause outbreak of in the susceptible wild and the domestic ducks. And as I told, this causes the duck viral enteritis, and which is also known as duck plague. And this disease occurs in domestic and wild ducks and other waterfalls, but this is not reported in other avian species other than the ducks, geese, swans, and the waterfalls. Even it is not reported in the humans and the other mammals. And in ducklings and the domestic ducks, this has been reported from the 7 days of the age to the mature breeders. And in ducklings of the age group of 2 to 7 weeks of age, losses may be lower than the older birds. And migratory bird is the major contributor to the spread of the disease, and ingestion of the contaminated water is believed to be the major mode of the transmission. But this virus may also get transmitted by the contact, and the virus replication takes place in the digestive tract, and then it moves to the bursa of Fabricius, thymus, spleen, and the liver. And the incubation period for the disease is 3 to 7 days. And this virus causes huge economic losses due to the acute nature of the disease, and it is having a high morbidity and a wide range of the morbidity and the mortality, and the mortality may range up to the 5 to 100%. So this, due to the mortality, also causes the economic losses, and it requires the condemnation of the carcasses. And also this disease is reported to cause the decreased egg production and the hatchability, which in turn also causes the economic losses to the industry. Coming to the clinical symptoms, this disease causes the sudden and persistent increase in the flock mortality, and this is often the first observation which is noticed in this disease. And other symptoms are anorexia, listlessness, nasal discharge, ruffled dull feathers, adherent eyelids, photophobia, and the extreme thirst. And ataxia may lead to the recumbency without stressed wing and the head with the head extended forward. Tremors, watery diarrhea, and the soil winds also can be seen. And often it is found that the sick wild ducks often they conceal themselves and die in the vegetation at the water edges. So these are the symptoms, clinical symptoms, like depression and the mild dyspnea, birds can be found lying down and dying from the duck plague. It may also cause the lameness, and also it causes the ocular discharges. And this disease is can be found in the both sexes from the varying age groups, and the as told that this disease is characterized by the sudden death, and apart from this, these causes also causes the lesions in the form of hemorrhages, progressive degenerative changes in the parenchymatous organs, and it causes the multifocal ulceration of the mucosa of the gastrointestinal tract. Whereas the multiple pale foci of the necrosis can be found in the liver and the spleen, and causes the lesions in the lymphoid organs. So huge mortality can be reported, and this can be diagnosed. So these are the lesions seen in the case of this disease. Here you can notice the enlarged and mottled spleen, petechiation of the liver can be noticed, hemorrhages and the diphthetic membrane of the esophagus, and this picture shows the mottled thymus, which is having petechi and the necrotic foci on the thymus. Diagnosis. Although the clinical finding and the lesions are the suggestive for the diagnosis, and this disease also reports the presence of inclusion bodies in the tissues of the affected birds, and this so the diagnosis can be confirmed by the virus isolation and the detection by the serological and the molecular test. And for this, the virus can be isolated from the specimen of the liver, spleen, kidney, and the intestine. And virus DNA can be detected by the PCR. For the virus isolation, 1 day old Muscovy or white Peking ducks can be used, and the inoculation can be done by the chorioallantoic membrane of the 9 to 14 days old embryonated duck eggs. And after inoculation, lesions can be found on the chorioallantoic membrane, and also the embryo, and hemorrhagic cam of the duck embryo can be seen. Here, hemorrhagic reddish duck embryo can be seen, and this disease must be differentiated from the other diseases like the hepatitis caused by the picorna virus or the astrovirus, also from the Newcastle disease and the avian influenza. For the control of this disease, live attenuated virus vaccine can be used in the commercial operations, and strict isolation of the infected flock must be there. Exposure must be prevented from the wild waterfall and the contaminated water, and the screening of the new stock should be performed to prevent the disease. So this disease is an important disease of the ducks, and can cause mortality, and this concludes the topic duck viral enteritis.